So what is the process of gastrulation? Now this process of gastrulation is an event involving a massive reorganization of cells which happens in the late blastocyst region. The earliest step, which is not really a process of gastrulation but something which precedes gastrulation, is where we all saw that inside the blastocyst there was a lump of cells which is called the inner cell mass. The inner cell mass will organize itself into two distinct layers. One is called the upper epiblast and the lower hypoblast, and this is one of the first steps in the process of gastrulation. So this upper epiblast and lower hypoblast, that is the two layered structure that is formed from the inner cell mass in the blastocyst, is referred to as the embryonic disc. It is sometimes also referred to as the bilaminar. Bilaminar means there are two layers. So the two layers of cells include the upper epiblast and the lower hypoblast, and that's why it is called a bilaminar embryonic disc. And all of you remember, during gastrulation the three major germ layers are established: the outer ectoderm, the middle mesoderm, and the inner endoderm. However, the first germ layer to be established during gastrulation is the endoderm, which happens to be the innermost germ layer of a developing embryo. Okay, so let us see how exactly the inner cell mass transforms into a bilaminar embryonic disc. So this is the blastocyst, what you can see here. This is the trophoblast, and see, this is the inner cell mass. It's labeled over here. See how it is just a lump of cells initially to start off with. And now did you see in the next step? In the next step, what happens basically is this lump of cells, which is the inner cell mass, has organized itself into how many layers? You can see here. It has organized itself into two distinct layers. The upper layer is made up of tall columnar cells. It is called the epiblast, and the lower layer has flattened cells to cuboidal cells, which is referred to as the hypoblast. See how clear it is. Here it is just a lump, but now here you can see that the inner cell mass has organized itself into two distinct layers. So this is what I was calling as the bilaminar. Now you know why it is called a bilaminar structure, because it has two layers. So this is what is referred to as a bilaminar embryonic disc. Correct? A bilaminar embryonic disc with two layers. Now very soon, let us see what happens. So let us follow the arrow. So after that, after this diagram, it is this picture. So this will be A, this will be B, and this will be C. Yeah, it's already shown there. So now see what happens above the epiblast. The upper layer is the epiblast, if you remember. This is the epiblast. Above the epiblast, notice a cavity has appeared, and this cavity that appears above the epiblast is referred to as the amniotic cavity. And also you can see the roof of the amniotic cavity is very soon lined by a layer of cells which are referred to as the amniogenic layer. The amniogenic layer comes from the trophoblast. See, this layer is the trophoblast. The trophoblast is the one that gives rise to the amniogenic cells, which occupies the roof of this cavity above, and this cavity above the epiblast is referred to as the amniotic cavity. And what is the name given to the cavity below the hypoblast? See, this is the hypoblast layer, and below that, this cavity is simply referred to as the blastocele. However, very soon the cells, what do you call these cells? Now these are the cells of what? These are the cells of hypoblast. I told you the lower layer of the bilaminar embryonic disc is referred to as the hypoblast. Very soon the hypoblast cells will start multiplying, and they will start spreading along the inner wall of the blastocele. See, these cells will multiply and they will start forming a layer. See, here you can see the layer. I'm highlighting it. This is a layer that is formed by the dividing hypoblast cells, and this layer is now referred to as the endodermal cells. It is called the endodermal region. See, endodermal cells of, in the future, this cavity. See, until the previous stage, this cavity was called a blastocyst, but now, sorry, a blastocele, but now this cavity becomes known as a primary yolk sac. So why has it changed from blastocele to primary yolk sac? Because its lining is changed. And where did this new lining come from? This new lining came from the multiplying cells of the hypoblast. Can you all see that this structure looks like the number 8? So it's as if, see, if I show the epiblast here, the epiblast is like this. Now let me show hypoblast. The hypoblast is also like a layer, and now let me show the amniogenic layer. It is like this. And now let me show the endodermal cell layer. It is like this. So it is as if it is like the numerical 8, isn't it? It's like the numerical 8 because you have a cavity above, which is the amniotic cavity, a cavity below, which is the yolk sac, and this layer is the hypoblast. Let us call this layer as the H layer, and this layer is the epiblast. Let us call it as the E layer, and this layer is the amniogenic cells or the amniogenic layer AL, and this is the yolk sac endoderm. We shall call it as YS, yolk sac endoderm. So it resembles what? It resembles the the numerical 8. So this is something you need to remember. It is much more prominent in the next slide. See how it is looking like 8, isn't it? See, numerical 8. Yes or no? Now you can notice how this entire 8 like structure, the 8 shaped embryo, is pushed further deeper inside. Why? Because a new tissue develops in between. I'm going to shade the tissue that is formed in between. The tissue that is formed between the outermost layer. What was the outermost layer? This outermost layer was the trophoblast. Even in the previous stage we saw outer to the 8 structure there is a trophoblast. So there is a new tissue that forms between the trophoblast and this 8 shaped embryo, and this new structure is referred to as the extra embryonic mesoderm. See, I've shaded it over here, but now I'm splitting the shading into 2 parts because very soon the extra embryonic mesoderm splits into 2 layers, a layer inside and a layer outside. Notice how the 8 shaped embryo is further shrinking in its size, and then here it was a continuous extra embryonic mesodermal layer, but now it has split into an outer layer which is called the somatopleuric extra embryonic mesoderm, and an inner layer which is called the splanchnopluric extraembryonic mesoderm. However, did you notice above this 8 shaped structure there is extraembryonic mesoderm still intact? It has not split. So at one portion of the developing embryo there is a contact between the outer layer, that is the trophoblast, and this 8 shaped structure, and there is extraembryonic mesoderm which has not undergone any splitting. But in the remaining parts, as you can see, a portion of it gets restricted to the outer surface just inner to the trophoblast, a portion of it gets restricted to the inner surface just outer to the endoderm layer of the yolk sac. So this layer which is close to the endoderm of the yolk sac is called the splanchnopluric extraembryonic membrane mesoderm, and this layer that is inner to the trophoblast is referred to as the somatopleuric extraembryonic mesoderm. Okay, so this is how the embryo, that is the inner cell mass, transforms itself into an embryo or into a very early stage of the human embryo, where you see this 8 shaped, please remember the numerical 8, the numerical 8 shaped structure lying inside the trophoblast, and very soon a tissue called extraembryonic mesoderm arises between the 8 shaped embryo and the trophoblast, and therefore it later gets split into an inner layer which is the splanchnopluric layer and the outer layer which is the somatopleuric layer, and there is a space between the two, and this space is referred to as the extra embryonic coelom. So this is a very quick review of the early stages of the gastrulation or the development of blastocyst.